Thank you everyone for joining. Hi everyone, thank you for joining us today. As Florence mentioned, it's a webinar on Jellagel as a next generation alternative to Matrigel. My name is Shante. I'm part of the commercial team here at Jellagel, and joined from our team today is our founder and CSO, Andrew Mearns-Spragg, who will be talking through the data shortly. To start us off, obviously our name is Jellagel, a revolutionary platform for regenerative medicine. We are also ISO 13485 certified. For a small company, this is a massive achievement as it gives all of our customers peace of mind that our products are certified for medical device uses. We were founded in 2015 by Andrew, a Scottish marine biotechnology pioneer who was surfing one day off the coast of Scotland when he came across blooms of jellyfish. This got him thinking, and he started doing some early research into using jellyfish as a source for biomaterials. Fast forward to where we are today, we currently operate out of our Cardiff office and manufacturing facility using the Welsh coast to harvest our jellyfish. Our next generation collagen type 0 is a breakthrough innovation in the fields of cell culture and regenerative medicine. Oh, my screen has frozen. I do apologize. Oh. Thank you. So the importance of collagen. Collagen is the most abundant protein in the human body. Jellyfish are over 600 million years old, meaning that their collagen outdates any other modern sources. So other animals have evolved due to environmental factors, whereas jellyfish have remained relatively the same from the beginning. This means that the jellyfish collagen that we use for our products is homologically similar to other types that are on the market. The problem: Matrigel has been the industry standard for cell culture for many decades, but recently supply issues, ethical concerns, and usability have caused problems for researchers. Matrigel is derived from a sarcoma that is artificially implanted in mice, meaning it is therefore chemically complex, and this can lead to batch inconsistency. The recent global shortage means that it's caused many delays on projects, costing customers and operations money and time. Jellagel provides an in vitro to in vivo solution, which is not possible with Matrigel. At the heart of all our products is our collagen type 0. As previously mentioned, jellyfish are over 6 million years old, which means that their collagen is a lot older than other ones available. This can be classed as the stem of all collagens. The benefits of our collagen type 0 are as follows. Obviously, it's non-mammalian as it's derived from jellyfish. It's biochemically simple, meaning there are no unwanted growth factors or biological contaminants that could negatively impact or influence your cell cultures. Batch-to-batch consistency in terms of collagen levels are near perfect, meaning that if you were to your results will be reproducible. It is also ethically derived and inert and cleaner at microRNA level. How does it compare? Well, our collagen type 0 compares to Matrigel. Obviously, the composition, as mentioned, collagen type 0 from jellyfish is different to obviously Matrigel, which is derived from a sarcoma in mice. It means it's biochemically complex, preventing you having less control over your cell properties. Also, usability. Our product doesn't require temperature control, meaning there is no need for a cold room or for it to be kept on ice or using cold tips during your procedures. Our product range, we have 4 main products. We have our research-grade jellyfish collagen, which is used for general cell culture. We have our coated plates, perfect for 2D cell cultures. Our 3D scaffolds used for tissue engineering and 3D cell cultures, and our Jellagel. Our Jellagel has all the same benefits as our collagen, and it comes in a standard size of a 10 ml kit, which comes with its own cross-linker and buffer. We have recently launched our sample 5 ml kits, which are a single-use version of our Jellagel kit. This is available and in stock at Clean Sciences now. I will now pass you over to Andrew, who will go through some data on our Jellagel.
Thank you, Shante, for that wonderful introduction. So it's a pleasure to talk to you today about some of the data, exciting data that we're generating using Jellagel for 3D cell culture. And just before I start, Jellagel essentially is a very easy to use kit, as Shante said, very user friendly and very simple to use at room temperature without the need for keeping any of your equipment or solutions on ice. But as we can see here, we've compared the performance of our hydrogel matrix Jellagel for comparing a range of cell lines. This is an example in human mesenchymal stromal cells, looking at competitor evaluation against Matrigel. And what we basically see here under the confocal microscopy results, you see that Jellagel performs incredibly well when it comes to the proliferation and migration of mesenchymal stromal cells. And at day 7, although that Matrigel will culture MSCs, you need to do additional steps to get the MSCs to become embedded into the gel matrix, which you don't need to do with Jellagel. It's a much more simple, easy to use and reduced steps in terms of getting to the end result. So it's a much more quicker, easier process. But as we can see, MSCs in this material are successfully supporting the growth of those viable cells. Again, this slide really just says that, again, Jellagel is non-toxic. When we compare it to certain bovine collagen, we see very good viability of cells cultured in this biomaterial. It's important to look in further detail. Are those mesenchymal stromal cells healthy and happy? When we look at the phenotypic characterization of those cell lines, they are producing the fibroblastic morphology that you'd expect those cells to display, and over 7 days they get more fibroblastic in nature and more mesenchymal like. And again, it's important to know what biomarkers are being expressed, what influence does the matrix have, and we're very pleased that actually when we grow mesenchymal stromal cells in the gel matrix, the mesenchymal stromal cells are expressing all the biomarkers one would expect to see from MSCs. And again, that is batch to batch consistent as well, which is very exciting and encouraging to see. And then the photograph to the right there, you'll see them the morphology of those cells also looking excellent as well. We also have a very keen interest in the usability of this biomaterial and this gel matrix for the production of organoids, particularly cancer organoids. And we have evaluated the gel matrix compared against Matrigel for the culture of MCF7 breast cancer spheroid lines. And what we do see comparing to the Matrigel is, first of all, the cancer spheroids that are grown in Jellagel are, first of all, more uniform and regular sized throughout the matrix. And we also see with the staining a better robust cytoskeletal net framework around the spheroid itself, which we think will provide great benefits in terms of greater accuracy and drug modeling downstream. And when we look at co-culture as well, you can use the material to co-culture. We've done this in this instance, where we've added to our human-derived MSCs a primary cancer cell line for co-culture. And here we look at lymphoblastic leukemia cells, which are stained in red. This is just cultured in 3 days in the hydrogel. And we've seen that when you look at the calcium stain greening, they show the viable cells of MSCs and the anti-CD7 conjugated to the lymphocytes as well. So again, very exciting preliminary data for co-culture. Another example of a cell line that we've looked at is embryonic stem cells. And again, when you culture these in the 3D matrix, first, as we've seen before, you get a very robust and uniformly regular sized spheroid throughout the gel matrix. But when you stain them with, in this case, alkaline phosphatase, they are consistent with the maintenance of their phenotype as well, which is important. We were very proud to enter into an OEM agreement with a exciting Boston-based company called Solaria late last year. Solaria are developing cancer co-culture organoid models to patient-derived cancer cell lines. And they are very excited about including collagen type 0 hydrogel matrix into their models. And where we've seen great results looking at organoids, growing 3D organoids of both pancreatic, breast cancer, and ovarian cancers with the necessary drug kinetic profiles associated with them as well, and where we see very good performance of the cells growing in our matrix. What we've seen as well, taken that to a T cell expansion model with Celaria, and in the photographs you can see the dark shadow in the top photographs are the actual cancer spheroid, and then what we've seen, we've seeded them with T cells, and what you're seeing here, the models of the organoids of the T cells expanding and then migrating and proliferating within the organoid itself, and then the bottom green looking organoids, that's with the addition of mesenchymal stromal cells as well, and you can see them migrating into the organoid as a little mini tumor. So it's very exciting work that these guys are doing, and their feedback has been very encouraging, where based on their results and their work on developing 3D organoid models, the collagen type 0 has minimal interference with tumor signaling, because unlike Matrigel, we have a reduced presence of biologically active motifs in the polymer, where Matrigel is full of growth factors and undefined. So that allows the researchers to focus on the cell-to-cell interactions rather than, and with minimal interference from the ECM, so allowing for greater accuracy and predictability of the cancer model itself. And thank you for your attention.
Thank you so much, Andrew, for going through the data. I will now open the floor to any questions from our viewers.
If you have any question, you can unmute your microphone or write on the chat.
Yes, hello, I'm Isabelle de Marcoulon. I'm from the Museum of Natural History in Paris, France. I'm very interested in testing this Jellagel as a substrate for growing or establishing primary cultures of marine invertebrates, especially corals. So I've been following the presentation. I have a question regarding the applications that have been that you have tested with this substrate. Did you try, non-vertebrate cell culture? Did you try aquatic organism cell culture? And the other question is, what exactly is collagen type 0? I didn't really quite understand.
Okay. Hi, Isabel. Good to meet you. The first question is, no, we haven't evaluated in non-vertebrate cell lines, so this would be a first for your team to do that. And secondly, we label it as collagen type 0 for the fact that, unlike because of the source, jellyfish are very ancient. They're 600 million years old, and the collagen that evolved to coalesce those cells to the jellyfish had to do multifunctional properties. So the collagen type 0 is a multifunctional collagen, where as collagen got more complex, as life got evolved, more complexity. So collagen type 1, for example, has evolved to specifically be associated with vertebrate biology, so bone, skin, and tendon. Collagen 2 is associated with very cartilaginous tissues, and collagen 4, for example, is one of those basement collagen membranes. The more physiologically simple an organism, the collagen had to do much more functionality, much more functional properties of that collagen to maintain cellular interactions. So collagen type 0 is our way of explaining that to the wider audience, where our collagen 0 has similarities to collagen type 1, but it also has demonstrated similarities to collagen type 2, 5, 9, and potentially others as well. Okay. And another question: do you have a simple, protocol that is visual to prepare this gel from the solution from the pouch to prepare it in the lab? Do you have some visual protocol that we can follow? Yes, we do. We have a video on YouTube. There's when you order the material, we can send you all of the instructions and videos of showing how to use. It's a very easy to use protocol procedure. I think it's about 4 or 5 steps. That's it.
Thank you.
I have a question. I'm Balou Jean-Marc from Crangin. The question is, could we functionalize your jelly gene to be used as a matrix to perform laser printing scaffold?
Yeah, so bioprinting is a very exciting area, something that we are looking at developing Jellagel as an ingredient to incorporate into inks, for example. And we haven't explored that in any great detail yet. It would require some research from the customer, but blending it with other materials that are inkable or printable would be a very interesting avenue to explore. It's something that we're doing ourselves as a research project anyway.
Okay, thank you.
We have a lot of questions on the chat too, so I will begin by the first one. Have you tried growing human intestinal enterites in Jellagel?
Not at this stage.
Okay, the next one.
Okay, can I excuse me? Are they endothelial like? Just I'm interested. Can you ask that? Is that yes or no? That particular cell line, is it endothelial like?
I just have this question. Maybe
the
the person
question.
who has the question can unmute the microphone and explain maybe more the question. No, maybe the person. Yes.
Yeah, sorry. Could you repeat what you were asking?
Yeah, I was saying is that cell line endothelial like out of interest?
Wait, I'm sorry, I'm hearing noises near me.
Is that cell line endothelial like in property?
So I'm talking about like human intestinal cells like jejunum or duodenum coming from patients.
Yeah, so we haven't tested those specific cell lines, but if they are endothelial in nature, we have got some very interesting data on endothelial cells.
Okay, thank you.
So, okay, the next question is any issue advised to use it in vivo for the establishment of orthopic lung cancer models?
We have tested this material in vivo. There's an abstract going to be coming out shortly, and as soon as it is out, I will share that with Denise. But it goes into some detail of the use of Jellagel in vivo in a certain model. But so the simple answer is yes, it has been in vivo with some very good results.
Okay, thanks. The next one, do you have any data on tubulogenesis on endothelial cells, for example, like UVEC perform on this collagen?
Yes, so customers have tested endothelial. I was saying that's my previous comment. We have had customers test endothelial cells in our gel and have generated some very, very good data in terms of the differentiations, tube formation assays, and things like that. So the answer is yes. I think I would recommend certainly the use of our material for those endothelial cell lines. I think that gives us some very good information, good data.
Okay, the next one, for how long the Jellagel is stable with plates, when plates with organoids, it degrades with time.
Can you repeat that, please?
In fact, it is a question about the degradation of the Jellagel. How long can you keep the Jellagel in plates with the organoids?
Well, we try up to 14 days, but possibly longer as well, depending on the certainly longer than 14 days.
So there is another question on the 3D bioprinting, but you answer, I think. The other one is about if it is possible to make dome with Jellagel. What is the G modulus? Is it only type 0 collagen?
So yeah, the dome assays are possible. The approach of dome assays are possible. The elastic modulus of the material is Matrigel-like, i.e. sub half a kilopascal in rheology. And again, the technical team here will be on hand to guide you through that process.
Okay, the next one is, do you have established and standardized protocol for passaging spheroids and organoids grown in Jellagel?
Yes, so there are, I mean, we can, so for example, you can release the spheroids through liver-rays, so you could then passage again. Those protocols are available should you need them.
Okay. Have you performed comparison with Matrigel in migration invasion assay?
We have looked at migration and proliferation of cells within the matrix, but we haven't compared against Matrigel, if that's correct. That's the correct answer.
What about the protocols for Jellagel digestions when passaging organoids? Are they the same for traditional than for the traditional Matrigel? Can Jellagel help in maintaining tumor microenvironment in patient-derived organoids from cancer?
So we use liver-rays as a standard protocol to release cells from the matrix without damaging the cells.
The next one, at which temperature Jellagel is solidified, jellified?
Physiological temperature, 37 degrees.
Have you tried growing melanoma cells lines in Jellagel?
Sorry, which cell lines?
Melanoma cell lines.
Melanoma. No, we haven't. That's one cell line we haven't tried.
Okay. What about collagen 0 organization? Does it form fibers like collagen type 1 and 3? Is it a substrate for collagen modifying enzyme like transglutaminase or lysis oxidase?
Okay. So collagen type 0 is a fibrillar collagen. In the process of creating the gel, it becomes fibrillar in nature. So it's very similar to type 1 in that regard. The second part of that question, in terms of, sorry, can you repeat the second part of that question?
So the question is it a substrate for enzymes like transglutaminase?
Glutaminase. We have actually cross-linked it with transglutaminase in the past, so there is potential there for that, but we've not looked into that side of it in any great detail as a substrate. But it's fibrillar, but we haven't looked into its substrate abilities.
Okay, thanks a lot. So it's all the things concerning the questions on the chat. But if you have any question, you can unmute your microphone and ask directly your question.
I have one question regarding PBMCs. Did you try your Jellagel to evaluate the migration of PBMCs from one compartment to another?
Not any great length, no, not yet. That'd be further work that people who've got to do that, would certainly be able to do.
Okay, thank you.
Okay, I don't know if there is other question. If there is no other question, we'll finish the webinar. So if you have other questions, don't hesitate to contact Clinical Sciences Group. So you have the email address here, and you can contact your local subsidiary. So we have different subsidiaries, but if you use this email address, you will have an answer, don't worry. For the questions concerning the protocols, the videos, etc., we will share all the things after the training. We record the training, so you can find the video just after this webinar. You will find the video on the Clinical Sciences website and the YouTube channel. So thanks a lot for the presentation, Shante and Andrew, for your time. Thanks a lot, everyone, to attend this webinar. And don't hesitate to contact us if you want to try the Jellagel. We have the possibility to send you some samples if you want to try. So don't hesitate to contact Clinical Sciences Group in order to try and to have more information about the Jellagel. This wonderful product. Thanks a lot, everyone. Have a nice afternoon.
Thank you.
Thanks again, everyone.
Thank you.